Meet the varicella zoster virus, the virus that causes both chickenpox and shingles. This devious virus belongs to the herpes virus family and has a unique structure. Encased within a liquid envelope is a linear, double-stranded DNA genome. Surface glycoproteins, such as GB and GH, facilitate its entry into host cells, where it hijacks cellular machinery to replicate itself. Initially, the varicella zoster virus invades the body, typically during childhood, causing a contagious disease called chickenpox. After the initial infection subsides, the virus retreats to the nerve cells near the spine, where it remains dormant for years, hidden from the immune system's radar. But lurking beneath the surface, the virus bides its time, awaiting the right moment to strike again. Factors such as aging, stress, or a weakened immune system can trigger its reactivation. When this happens, the virus travels along nerve fibers, manifesting as a painful rash along the affected nerve pathway. This condition is known as shingles. Shingles can appear anywhere on the body, but it typically stays confined to one side of the face or body. Burning pain and itching are early signs of infection. A person infected with shingles would have a painful rash or small blisters on an area of skin and may also have a fever, headache, muscle pain, and nausea. Now let's look at our immune system as it confronts this invader. First responders in this battle are the components of the innate immune response. Macrophages, neutrophils, and dendritic cells detect the presence of the virus and swiftly mount a defense. Toll-like receptor 2 recognizes the molecular patterns of the virus and sets off a cascade of inflammatory cytokines, most notably interleukin-6. Macrophages that were activated by TLR2 signaling then engulf the virus-infected cell. Most of the time, barrier defenses are already compromised since shingles resurfaces in people who have already had chickenpox. Next, we enter the adaptive immune system with its arsenal of highly specialized cells and molecules. Upon recognizing the viral antigens presented by dendritic cells, T cells spring into action. Cytotoxic T cells target and destroy virus-infected cells, preventing the spread of the infection. Meanwhile, B cells undergo a transformation, maturing into plasma cells that produce antibodies specific to the varicella zoster virus. These antibodies tag the virus for destruction by other immune cells and neutralize its ability to infect healthy cells. As the primary immune response subsides and the infection is cleared, a select group of memory cells remains vigilant. These memory T and B cells essentially remember the varicella zoster virus, ensuring a rapid and robust response upon re-exposure. This way, if the virus tries to resurface in the future, the immune system is able to launch a swift and powerful secondary response. Memory T cells recognize the virus immediately and coordinate a rapid deployment of effector cells and antibodies, effectively containing the infection before it can cause widespread damage. But is there a way to arm our immune system against the varicella zoster virus before it even encounters it? Yes, through vaccines. Vaccines are a powerful tool in the fight against infectious diseases. They contain weakened or inactivated forms of the virus, allowing the immune system to produce a response without causing illness. This way, our immune system learns to recognize and mount a defense against the varicella zoster virus, significantly reducing the risk of both chickenpox and shingles. Vaccines are a perfect way in which we can harness the power of our immune system to prevent disease and protect public health.